As an introduction, my name is David Tan. I'm the co-chair of the TPLG Pediatric workstream. I'm working for AbbVie as my full-time job. So today I have the pleasure of introducing our friends from the European Pediatric Formulation Initiative, short for EuPFI, Dr. Smita Salunke and Mr. David Clapham. Our first speaker, Dr. Smita Salunke, is the Chief Scientific Officer and Chair of the excipient workstream for EuPFI. Dr. Salunke has extensive experience in pediatric formulation and is the creator of the STEP database. And she is also critical in facilitating the strategic direction for the EuPFI Consortium. Our second speaker, Mr. David Clapham, is the chair of the Taste and Taste Masking Workstream in EuPFI and has over 40 years of experience. He recently retired from a major pharmaceutical company and is now serving as an independent consultant. Without further ado, let us welcome Dr. Smita Salunke and Mr. David Clapham.
Thanks, David. Thank you very much for the kind introduction and giving us this opportunity to present what EuPFI is, what we do, and probably highlight some of the areas where EU and EuPFI and IQ have the common interest and where we can work together. So, yeah, thanks again for this opportunity. So, for the introduction, what I would do is I'll go through the quick facts on what EuPFI actually is. So understand we all appreciate the importance of the development of safe and effective medicines for children. And now the changes in the regulation has changed the way we develop the medicines for children. And the pediatric drug development was done before. And today the pediatric drug development, I think, is integrated into the drug development. And we face several challenges when we develop the pediatric formulations. And these challenges are somewhat kind of shared by the many stakeholders within the industry, academia, hospital, and the regulatory agencies. So this is where the EuPFI actually came into picture. The EuPFI was created informally first by the 7 founding members, based on a very genuine willingness of formulation scientists to work together in a non-competitive environment to understand how the formulation research and development for the children could better fulfill the needs of sick children. And this is how it kind of, the group was formed and it was started. But slowly down the years, it has now evolved into a very structured, established consortium with a mission to promote and facilitate the development of safe and better medicines for children. So this is our 10th anniversary. It was established in 2007, and it's 10 years down the line. We have members from industry, academia, hospital, and regulatory agencies, and we have 5 work streams which are centered around the 5 pediatric formulation initiatives. So, as a part of promoting and facilitating the research in pediatric formulation, we organize annual conferences as well. So far we have done 9 annual conferences, and me and David today will take you through the different work streams that we work on, what are the objectives of this work stream, what our current priorities are, and then give you the snapshot of the conference as well. Okay, moving to the next. So this is the EuPFI members. We have 10 pharmaceutical companies: AbbVie, Astra, CleanSearch, GSK, Janssen, Lilly, MSD, Novartis, Roche, and Sanofi. 4 associations. We've got 8 academia, and the sponsors with Pfizer, Bill and Melinda Gates Foundation, GREEP, and then the USPFI, which is NICHD, and then Pyramid Healthcare. And mainly the EMA acts as an observer to the group in a passive way. They contribute to the exchange of the comments and the recommendations raised by the group and so on in a very positive and productive way. Okay, and this is kind of to show you the EuPFI framework on how do we work. So we have different levels of membership options and the criteria. So the way in is that the EuPFI membership comprises of EuPFI members, EuPFI associates, EuPFI sponsors, and the observers. And then we have 5 different work streams: devices, age-appropriate formulation, bioform, excipients, and test masking and test assessment. And the overall group meets face-to-face twice in a year, while the monthly TCs are organized to discuss the day-to-day or the regular issues and the issues within each work streams. And for the individual work streams, the monthly teleconference is organized. And as I said, we have the annual conferences as well. Okay. There you go. So that's the annual conferences. As I mentioned, we have so far organized the 9 annual conferences, and this year it will be our 10th anniversary conference, which will be held in London. I'm trying to move. Okay. Probably that should work. Yeah. So this year we will have our 10th anniversary conference, and that will be in London. So as usual, we will have the pre-conference workshops. There are 2 workshops which are organized. One is the plastoboceutical, where I will be discussing the formulation challenges doing a workshop on formulation challenges in pediatric clinical trials. And then the second workshop is the excipients, which is organized by the excipients workstream members, and it will be on determining the best practices on the selection of excipients for pediatrics. We also have the sub-box sessions, poster sessions, exhibition, and innovation showcase, where the different manufacturers they present their innovations, and then we have the case studies presented by the industry pharmaceutical companies as well. So this is the snapshot of the 5 work streams, each addressing a fundamental aspect of developing medicines for children. And my colleague David will take you through the each work stream on what are the current priorities and the objectives of this work stream. And what we would do is we will mainly kind of elaborate on the test masking and the excipients work stream to kind of demonstrate on how we collaboratively have worked with IQ and are working with IQ and the other stakeholders in this field. So over to you, David.
Thank you, Smita.
Oops, sorry.
Okay, so as Smita said, we will give you a snapshot of 3 of the work streams and then 2 of them in a bit more detail. The 2 that one is Smita is the chair of and the other I'm chair of. So we are looking at age-appropriate formulation group, and in this case, it's not just pediatric focused, but geriatric also, so age-appropriate in all forms. What we're looking for here is to provide guidance to industry, regulators, academia, of how to ensure that we have age-appropriate formulations. So also considered within the pediatric sphere, the various age groups, to make sure we are appropriate to those age groups. So there is work going on in looking at the dosage form acceptability, safety, efficacy, and dose flexibility of, for example, tablets, liquid dosage forms, and so on in the orals, looking also at injectables, inhaled, basically all the different routes to see if we can provide guidance here on making sure we tailor the dosage form to the patient group. A particular challenge is modified release formulations because we've got different physiology, and looking at how that can be taken into account in age-appropriate formulations, but also taking into account that children may need modified release formulations so as not to, example, need to take medication during their school day, which might make life difficult for them. So there's routes of administration, looking at the various options we have there. A particular challenge is the interaction with food and drink, sometimes deliberately, where we're trying to give that the dosage form in food or drink to taste mask, or to make it more palatable, more easy to dose, but also inadvertently, if you like, when food is given with their normal daily meals. and then looking at the effect of co-administration. So it's quite a large area. There have been a number of papers that we have been prepared, a lot of it in International Journal of Pharmaceutics. I won't insult your intelligence by reading these, but what we've looked at is things like formulation factors, how they affect the acceptability of different oral medications, looking at compounding and manipulation issues, so where we need to subdivide dosage forms, for example, and how that works, and then looking at the benefit risk approach to selecting an appropriate pharmaceutical dosage form. So accepting that there are things that need to be balanced, like efficacy, safety, but also patient access, because sometimes we do need to think about the ability of people to pay for these medications. And some more recent papers. So this group also has the challenge of looking at the acceptability of oral formulations. So that's distinct from palatability, which is what my group does. Looking at safety and access, and looking at things like swallowability. There are a number of ongoing work papers in preparation, so this industrial perspective on pediatric formulation, which is a collaboration with IQ. Strategy for developing poorly soluble drugs for pediatric delivery, where the lessons learned from poorly soluble drugs in adults have to be tailored to children, and then looking at food effect. And you'll see as we go through this, there is some overlap between what the groups do, but then we do collaborate with each other in the groups as well. And we just jump on, sorry. So the Royal College of Surgeons in Ireland have now got a grant application for 4-year studentship to look again at admixture with food. And in terms of assessing the acceptability of tablets in children, there's a work stream looking at 3D printing, so that we can look at things like size, shape, color, but also surface characteristics. How rough is the surface, how smooth, what do you coat it with, and so on. So a lot of interesting work going on there. Another work stream is looking at administration devices, so looking at the way that we now give that dose. So particularly interested in oral dosing syringes and how we can specify those, because there are no international standards on the oral dosing syringes. The current control is purely by parenteral syringes, so needing to look at that and what we can do. Also understanding how people actually use these things in practice, as opposed to what we tell them to do. So again, there's a couple of papers looking at the currently available devices and the challenges. Also asking people who actually use these things what they think of them and what they would like. And in the ongoing work sphere, we've got a survey, and there's been a pilot study to investigate what questions we should be asking in order to understand what parents and carers and children and so on make of the dosing devices they already have, what they would like to see, how we can make them better. And a little heads up for your diaries, there is to be a workshop in London, date yet to be decided, where we're going to look at the administration of oral liquid doses using oral dosing syringes and enteral accessories also, but for nasogastric tubes and so on, to try and come up with some agreed standards about how that should be done best. The final quick snapshot is for the biopharmaceutics group, and as you will all know, biopharmaceutics is an area where there is a lot of unknowns, particularly in the how the pediatric population develops, how their livers change, how their kidneys change, how their elimination changes, and so on. So this group has been set up to understand the current tools available to measure biopharmaceutical performance, to look at how maybe the availability classification system needs to change to take into account the pediatric populations, and highlight those areas where we really don't know what's going on, and to promote additional research into those areas. So again, there's been a number of papers already published where we've been reviewing the literature and trying to understand knowledge gaps, and then bring together some of the key issues in extrapolating from adults into pediatric populations, for example. So our current priorities in this area are to prioritize research activity to be undertaken in the pediatric biopharmaceutics sphere, to understand how pediatrics develop, review the strategies currently used in bridging studies, and review the impact of food. So again, another group that's looking at the impact of food, but particularly again, from this case, from the biopharmaceutics point of view. So all of those, that's a very quick snapshot. And now Smita is going to take back over and talk about the pharmaceutical excipients work stream. And then I'll come back and talk about the taste and taste masking one.
Thanks, David. So basically for the excipients work stream, the main purpose of this work stream is to address the safety and toxicity issues and the use of excipients in pediatrics as such. And as you may all know, the regulatory agencies do come back to, and everybody in the stakeholders are facing the challenge of the use of the excipients in the pediatrics. We have to justify what excipient we're using, the role of the excipient, the amount of the excipient that we're using, and obviously, the safety and toxicity data needs to be provided for the excipient that has been used or proposed in the pediatric formulation. However, there is very limited data available on the safety and toxicity in terms of the pediatrics. And as this limited data was scattered around the sources, it was difficult for the for industry members, for the stakeholders to find the information in the public domain. So the EuPFI excipient work stream suggested building a database where we could compile all this information together and have this information in one single source, so that it can be then looked into and if used for the selection of excipients, for submitting the data to the regulatory agencies and so on. And this is where the STEP database then came into picture and was developed. So the database was launched in 2014, and this database kind of, as I told you already, that it compiles all the safety and toxicity information, and the search interface in this database allows you to search by excipients. So if you already know what excipients you are looking for example, propylene glycol, you can just go and select from the list of the excipients propylene glycol and you'll find the information for it. But for instance, if you're not aware, you would like to see what excipients are used in children under certain age and what effect it has on the digestive system, then there is another search functionality that is available, which is search for excipients. And there you can, it's the search kind of interface allows you to select different options depending upon your query, and then you get the list of the excipients which are used in, as I said in the example, used in children under certain age and has the effect on the effect of the digestive system. The interesting part about this is that the results page in this database allows you to filter the data according to the age, to the route, to the duration, species, and so on. So even though if you have if you go and find the information on the propylene glycol and you come to the results page and you see that, okay, it's got all the information about the adults, children, animal, but I would like to see only if it has an effect in the children, then at this stage as well, you can go and just filter your data like you use your normal Excel sheet and just filter the data and find the and look at the information. So overall, the interface is made to be very, very user friendly, and it's being used by around more than 3,000 users now from the industry, academics, hospital, and regulators. And everybody appreciates the information that's available in it. We've got 3 publications on this database, and it is open access. So if you need any information about how to use this database and how did we came about building this database, you will find all that information in these publications. Okay, and it is already cited in more than 90 publications. So it has been used widely, and people find it very useful. So this is just to give you an idea on what sort of queries can you run on this database. So for example, if you were after, what are the findings for excipient X following an oral administration to children of all ages, then this is where you will find the information easily into the STEP database. For example, if you're looking into the specific query as in, is there any toxicity to skin caused by propylene glycol in children under 4 years, then again, with the limitations and the search, you can find such information. So this is an example, and feel free to go through all these queries on what type of examples and what type of queries the database will answer for you. Okay, one thing I definitely would like to mention here is the database, the information in this database is as is. So we go into the literature, find the literature, extract the information from the literature as is it is in the publication. So we don't provide any summary. We don't do any critical appraisal. It is just, the idea is to just guide you with the information for the further analysis. So you can take all this information and probably and provide it to your toxicology team to do the further evaluation. But we by no means do provide any summary on it. So it is the responsibility of the scientist to check and assess the data and the quality of the evidence before use, because we take it as it is from the publications. Okay, the database is available from EuPFI website. As I've shown you here, once you go into the EuPFI website, you'll find the pink box here, which is STEP database. If you click on the box, you will be taken onto the STEP database link, and you have to be registered to use this database. So as you can see here, there is the user guide and there is the new user register here. So when you register here, this is, again, it's free to use. The registration information helps us to know who all are using this database and who finds it useful. So yeah, so you have to just fill in the your name and organization details, and that's it. And you will get the email address and password to log in into the database. It's freely accessible to everyone. And there are the quick links provided over here, which will help you for further use of the database. So we've got the brochure, we've got a sample record to show you how the results page will look like, the list of excipients in the database. There are some known issues with the database which we are working on, but we have listed them here just for your information if you come across any of the issues while using the database. There is a feedback form, so feel free, it would be very helpful for us to get the feedback from you in terms of the usability. If you would like to be more user-friendly or anything, please do fill in the feedback form. There are video tutorials available as well to show you how to log in into the database, how to search by excipient, how to search for excipient, how to understand the results page, and so on. And then we publish the newsletter regularly as we put in more excipients into the database. We release the newsletter to let the community know what new excipients we've added into it. So that's it is. And then if you need any more information, feel free to call me, to write to me, or to Barbara from EuPFI. EuPFI, we have developed this database in collaboration with EuPFI, and they help us to find the information. Barbara is from NIH, so she helps us run the searches in the NIH, and she's an information scientist out there. So she's very well versed with how to search the information, how to run the queries, how to prepare the search strategy. So she helps us with all that for developing this database. So that's how we have worked with the with EuPFI in developing this database. And IQ has been has played a very important role in this STEP database. They have indeed contributed to the quality and quality assurance and the usability testing of this database. So yeah, there has been lots of collaboration. The work has been done in collaboration with many people. So currently the database is populated with 65 excipients, and we, on a quarterly basis, we update the data that's available in the existing excipients and the new data that is available to us. The list of the excipients, I've put them in the backup slides. So you'll find the list of the excipients currently in the database, and then the excipients that we are working on at the moment. Just for your information, Bill and Melinda Gates Foundation has sponsored 10 excipients, which would be useful in developing the medicines for children for developing countries, and we're currently working on those excipients. So here is just the to give you the idea that we have completed the work on the clinical, non-clinical, in vitro regulatory data, and now we're working on expanding the data content with more excipients. And there are some sources which are fee-based sources, or the company-donated data that is at the moment not included in the database, but we hope to include that in future. But this would all depend upon the funding available to the database. And then there are some wish list from different members on having the more information into the database, as such the API information, interaction data, having some additional fields or the links to the external sources, excipients combination data, and then the list of the excipients in the marketed pediatric products. This is the wish list from different members that we would like to add to the database. But at the moment, with the fundings that is available to us and with the prioritized scope of the database, we just have the clinical, non-clinical, in vitro data and the regulatory data in the database, and we'll see how it goes. And in future, if the funding is available, we'll go into the list that we have put here for the future of the database. So as I mentioned, the database as such was funded by EuPFI in the development, and we launched the database with the 10 excipients, and then it was slowly further funded by EuPFI to add on more 50 excipients into it. But at the moment now, EuPFI just maintains this database, and we still need to sustain the database. So we have come up with a scheme which is sponsor an excipient. So if any of the industry members or the excipient suppliers, if they would like to add any of the excipients which is not there in the database, but excipients of their choice in the database, then they can easily do that with this scheme. So they can sponsor an excipient. It means that we would be able to fund the services to get this additional resources to do the daily task of collecting and curating the information. And it's a win-win situation for both of us, but then because the stakeholder gets the excipient they want in the database, and so on, it's helpful to the community as well, because they get that information on the excipient in this database. And the amount is not too high. It is just between 2.5 to 5K. It depends upon the number of the references that we curate actually. And if it is too less, it could go to even less than 2.5K. So if you approach me, we can do the rough search for you and see how much it comes to. But please let us know if you need any more information on sponsor an excipient or an excipient you would like to add to the STEP database of your choice. So that is just to summarize on the STEP database, that it serves as a repository for safety and pharmacovigilance data for the pharmaceutical industry, related industries such as food, cosmetic industry, to help guide decisions. It provides a basis for assessing the need for generating new data for the pediatric medicines in order to clarify what kind of new data may be required. So you will be able to identify the gaps and then so on, the need for the new data. It provides a platform to share the data from manufacturers, industry, public, and government agencies. So please give us your feedback. I've given you the link there, and anything you need to know about the database, feel free to get in touch with me. And then another ongoing activity within the excipients workstream is the paper that we are actually working on at the moment to identify or determine the best practices in the selection of excipients for pediatrics. So everybody has to go through this process within the industry, and everybody has their own way of doing it, their own practice of doing it. So we thought that we would get together, see what are the different practices available or used within the industry in selecting the excipients for pediatrics, identify what are the guidelines available, are there any gaps in the guidelines, with few case studies from the excipients, and that is something we are working on at the moment in collaboration with IQ. So, and on the same lines, we are organizing a pre-conference workshop at the EuPFI 2018, which will be led by Kevin Hughes from IPEC, and it will be moderated by the excipients workstream members. It's on 11th of September. The main conference is on 12th and 13th of September, and the pre-conference will be on the 11th of September, and it will be in London, in UCL School of Pharmacy. So that's all from the excipients, and I'll give it over to David to take you through the test masking and test testing workstream.
Thank you, Smita. So yes, I'm going to talk about taste masking and taste testing. And this is an area which is very dear to me. since I've spent a long part of my working life trying to make medicines acceptable and taste better to people. And one of those areas that we might like to work on is to try to head the problem off at source by being able to test the taste of the API before it ever gets as far as a formulation. So what we are aiming to do in this group is looking at understanding the current state of the art of taste masking technology and taste testing technology. And to be fair, the main focus of the group at the moment is the taste testing area. We do have some work in the taste masking area too, but that is following on behind, if you like. We need to understand also the limitations of the technologies and how we might then be able to apply them. So there are some papers out there already that this group have produced. I've only been in chair of it for just over a year, so I'm playing catch up on some of these things too, but looking at the taste masking of poorly palatable drugs and look at the different approaches to taste masking. But as I said, also to look at how we might maybe avoid having that problem in the future. The reason for a lot of this work is that palatability and acceptability and palatability are not the same thing, but palatability is an important factor of acceptability. It's widely acknowledged as a major factor in compliance, especially in children. If you've ever tried to give a medicine to a child that it didn't like twice, you'll know that you may get it in once, but not twice. So it's very important to understand this and to make it palatable. But the issue about that is hindered by a number of factors, including firstly, poor understanding of importance, not maybe amongst the formulation scientists, although often even then, but particularly amongst management who say, if it works, where's the issue? But it's really important that medicine is palatable, otherwise it won't be taken. There's also a very poor definition of terms. Palatability and acceptability are used convertibly. They're not. Things like mouthfeel are not really understood. So it's very important that we define terms. There's also a lack of agreement of how to measure it, particularly in children. A lot of studies are done in adults, but children are not small adults. It's important that we understand how to do this, at least how we extrapolate the data from adults to children. And a particular area of lack of understanding is in the regulatory authorities. They do not understand the terminology we use, and it's important that we help them to do so. So as a result, there's a need for best practice guidance on this whole area. So this group exists, to support direct methodology development to evaluate the palatability of API and formulations. Will include human, but also non-human, in vivo and in vitro methods. So as I said, if we can add as a factor in the choice of an API what it tastes like, maybe we can help to avoid the issue, or at least know the magnitude of the issue we have, and then what we need to do to solve it. Particularly in the area of respiratory medication, for example, once you have chosen the molecule, if the first thing you hear of a bad taste is in a first time in human study, you have a whole world of trouble to go back and identify maybe a different molecule or a different salt, because there is nothing you can do really to taste mask an inhaled product. So we can look at that. The other thing we're looking at is to identify a test set of compounds to evaluate technologies. I'll speak a bit more about that. To support best practice in product development of palatable pediatric formulations, we all know that there's not that much guidance in pediatric formulation per se, but looking at the palatability of them is an area which is even less well served. Then to support development of taste masking approaches, including how to evaluate their applicability. And finally, but maybe most importantly, to support and direct fundamental research into understanding factors that affect the differences between pediatric and adult palatability. So we have a number of ongoing, more targeted work, which is addressing some of these. One that's in development right now is the assessment methods tool. There are a number of people who will tell you they can assess the acceptability and palatability of your product, but just how good are they? So what we are coming up with is a tool based around 12 R's that cover analytical factors, accessibility factors, and applicability factors. The first set of those is on the screen, and then what we're going to do is look at the applicability of those in 3 domains, looking at the API, how they might be used in helping to formulate things, and what's also important is looking at stability, because if APIs can be fairly unstable, flavors and so on which get used are even more unstable. They are essentially encapsulated volatile oils, and the volatile bit gives it away that they're not particularly stable. So the taste of a product can change markedly over time. One that I worked on was very nice when it was first made. By 6 months into the stability protocol, it had changed and become really unpleasant. So it would be nice to be able to monitor that by some means other than a human study, or even predict it. So we're at the stage of early testing right now, using just a few techniques that we have experts in the group, and using a traffic light system. So red as in poor, amber probably okay, green it's good at that, and leaving white where it identifies that actually we don't know, we maybe need to go and find out. So our aim is to make that fully quantitative in time, so we get a score for each of the factors, and then we can come up with clarity about what techniques are and are not good at. We also need then to test a wider range of techniques, and we will be coming to IQ members or to EuPFI members and 1 or 2 others to ask what techniques we should be evaluating. And particularly if you happen to be an expert in that technique, to help us to populate the tool. We'll then validate it by looking at other people who've tried to do something similar, and then make it available. I spoke about a compound test set. One of the issues around evaluating any technique, including human studies, is how we can compare data across techniques, across laboratories, across time. So what we are looking for is a well-characterized small set of compounds which are soluble, stable, readily available, consistent, so we can get the same thing year after year, and preferably publishable, which will make it easier for people to get funding for studies. Covering a wide pharmacophore space, so that we are looking at a whole load of different types of molecules with a wide range of bitterness, intensity, and character, because bitterness is not one thing, and character changes. A look at range of other sensory attributes like salty, sour, sweet, umami, metallic. There's a whole host of things that pharmaceuticals can be, apart from bitter, although bitter is the major issue. Looking at with human data in an agreed format, so there are quite a lot of data out there, but how we put all that into one format, and then validated non-human data, so preclinical models, for example, the rat Barter model, which is getting used more and more now. We see this as having used as a calibrant for human taste studies, so that we can compare data between labs and over time. Non-human taste studies, like the rat model or any other model you like to think of, but also for organoleptic stability studies, so we can calibrate over time, and also useful in method development for taste or other organoleptic factors such as trigeminal effects, mouthfeel, and so on. So this is a hope, but if you have suggestions for compounds that you think could be useful in such a compound test set, I would be very happy to hear from you. A final area we're working on actively right now is a guide to good pharmaceutical sensory analysis practice. So there is guidance for food and topicals, but it's poor for pharmaceuticals. There's confusion in the industry about what is and is not acceptable to do, and what is and is not acceptable data. And we are hoping there is scope to get regulatory buy-in to such a document, so that the quality of studies done to support the acceptability of a product, they will have a standard by which to judge that, rather than it being a bit hit and miss as it appears to be now. At the last conference, we did a workshop doing a sensory study on different T samples to show how the idea worked. And there was a lot of feedback that was very useful in understanding what was required, and an appetite for us to develop such a thing. So we're working on that right now. Oops. There are some direct collaborations with IQ right now. So we're both contributing to workstream activities on excipients and age-appropriate formulations, and also on this first transatlantic workshop on pediatric formulation development. And you can see some of the papers that have come out of that. And also on the WHO guidelines. So they have put together a research toolkit for drug development, particularly for antiretrovirals. But as you can see, one of those modules is acceptability, module 5. And WHO approached IQ and EuPFI as the subject matter experts in this area. So it's a very interesting and welcome development to be able to input to this toolkit. And together, I think we can produce a very useful document which will help people not just for ARVs, but any kind of pharmaceutical. And the other area we've been involved in heavily of recent times is the SPEED initiative. As you can see, one of those is in vitro taste testing. So that's looking at cell-based models, non-human models, and so on. And we've been involved in that. And particularly members at EuPFI have been heavily involved in that, and particularly members of my group. One question that's floating around the group is whether we should promote palatability, non-inferiority, as a criteria for assessing suitability of generics. We do assess bioavailability and quite a number of other things, but if a product is actually tastes much worse as the generic than it does as the innovator product, maybe there's scope for saying it should also taste at least as good as the innovator, in order for it not to be a disincentive to people using it. And hopefully that's something we will consider for the future. So that's my bit done, and back to Smita.
Thanks, David. Yes, so for me, what I would like to do here is just summarize. I've put up this slide to just summarize the key outputs from the EuPFI members. So we've got this 3 papers, which are the key papers for the STEP database from the excipients. And then from the test masking, there were some very useful papers which you will find. So we've listed out all the key and the useful papers that you may find useful on the slide. So feel free to go on it. And they're available on the EuPFI website as well. At least a link to and then the full text would be available to you depending upon the subscription. But yeah, and then the collaborative outputs as well. So you'll find all that on the EuPFI website as well. So just wanted to kind of, for the closing, reiterate the mission and the objectives of the EuPFI. So the mission here is to promote and facilitate the better medicines for children. And we would do this by sharing expertise, interactive discussion between the industry, academia, clinical, and regulatory professionals. We disseminate the information through the annual conferences, raise the awareness through the annual conferences and our publications. We organize the workshops as well. And obviously through the linking and networking with different stakeholders who are working in this field. The main objectives for us is to identify the issues and challenges and find a way forwards to address those challenges in developing medicines for children, promote the early pharmaceutical consideration for development of pediatric medicines, identify the potential information knowledge, know-how gaps in pediatric formulation development, and obviously improve the availability of the information of pediatric formulation. So that's kind of the objectives, and this wouldn't be possible without the team. So the success of this consortium, I would say, has been achieved by the balance between the shared vision of the consortium, of course, but the added value of each member that is there in this consortium and the aims of each work stream that we just presented. So you will find that we have an article on the EuPFI which kind of summarizes our and the lists our outputs and the key accomplishments, and you'll find all that in this paper. So we've summarized all that in this paper, and again there are the members from IQ as well who's contributed to this paper. So, and then there is a website, so that's www.eupfi.org. It has all the information about the memberships, on how you can join us, what are the different ways you can collaborate with us. The work streams updates are available on the website, conferences from the past conferences and what presentations we had. Unless you attended this conference, you won't get the access to the presentations, but at least you will come to know what are the presentations, and you can then at least approach the author if you want to access to the presentations. We also try to put in the new news and the links of the interest related to pediatric formulation on the website, and of course the EuPFI publications, as I told you, are all available on the website. So thanks to all our EuPFI members. I think so the consortium heavily depends upon the time and the commitment that is given by the members, and who have the conflicting priorities but still give their own time to the EuPFI activities, and really appreciate that. So thanks to EuPFI members, and thanks again to IQ for giving us this opportunity to present EuPFI, and thanks to all of you for your attention to listening to us. Thank you.
Thank you very much, Dr. Salunke and Mr. David Clapham. And with that, we're open for questions. Please key in all your questions into the question box. Well, I will start with a question for an excellent presentation again. I'd like to know a bit more on the STEP database, where you highlighted about the future path forward. And with regards to the API information interaction data, how do you plan on getting those information? Are you working with some pharma companies, or you're just obtaining more of a generic API?
No, David, we haven't actually started with this activity, and not even thought about how do we go on about it, because this is something, you know, when we did the need assessment study on what type of information should be included in the STEP database, it came up then that people would like to have this information in the database. But then as a part of this need assessment exercise, we had to prioritize on the information that we can put into the database depending upon the funding that's available to us. So the prioritized information was the clinical, non-clinical, in vitro data, and this information we thought that we would put into the database later on if we have the funding available to us. So to be honest with, we haven't thought about on how we would put this information, because we're not thinking of putting that into the database as of yet. So the discussion is not started on that aspect yet. It is something for the future. So
yeah, thank you. I think that will be very helpful, and I myself as well look into STEP database as well, and it's very well established, and thank you for all the good work that you have done on that.
Thanks, David. Really appreciate that.
And so the next thing I'd like to know more will be the taste assessment and taste masking stream. And I think so David gave a very good overview on the activities that you have conducted. So David, I know that in the past there was some work done to look at some in vitro testing using the electronic taste, e-tongue in short. So are you still working heavily on that for the future? I know you mentioned some of the methods that we'll be using, like MATA and some other cells to evaluate taste assessments.
Sure, yeah, so it is one of the techniques we will be evaluating. We have a number of people in the group who have used it in the past or are using it now. The summary feeling about what the technique is good for is comparing compounds side by side, and also looking at formulations in terms of whether formulation A is better than formulation B in terms of taste. But the technique is not good for comparing data over a period of time. So if you're wanting to compare a reading from today and 6 months ago, then it really does not do that particularly well, is the kind of a summary that people feel about them. There are several different kinds of electronic tongue as well. So the two that we've looked at are the Alphamos and the Incent one, but I know there are others available too. So it is something we will be looking at. So we are looking at those kind of in vitro techniques, also looking at cell-based methodologies, of which there are a number, and animal-based techniques, such as the rat barter. There's also a mouse barter, and there are a number of others. So that's kind of the focus of our efforts, if you like. But electronic tongue is being looked at.
Just curious, will there be any comparison or evaluation to a USP method using a modified USP1 or USP2 for taste purposes?
So is that taste masking and looking at how the taste masking exists, basically sort of dissolution, or is that using that as the? Sorry, say again.
So are you comparing methods using dissolution method and the E-tongue as well?
No, but there is work on that kind of out there in the literature already, and basically that says essentially that if the dissolution shows that the concentration of API available exceeds the taste threshold, then you will taste it, and how badly you will taste it will depend on how much over the taste threshold the API is. So dissolution is a useful method for monitoring how effective taste masking is and how long it lasts. You don't really need an E-tongue to do that. What the E-tongue will tell you to some extent is what type of taste you're going to get once the API is released.
All right, thank you. And I have one last question, and to Smita, with regards to participation in EuPFI. So can we recommend our attendees here to contact you directly if they are interested to join?
Yes, please. Yeah.
All right, sounds good. Then I think if there are no other questions, then I would like to thank Dr. Smita Salunke and Mr. David Clapham once again for taking time to share with us the work that EuPFI can help to make pediatric formulation better. And with that, I'll pass the ball back to Mahalia.
Thank you, David. Thank you also, David Clapham and Smita Salunke. I would like to thank all of our audience for joining us this morning for this very interesting and insightful webinar on what the EuPFI is and how they can help us going forward. Thank you again for joining us, and have a great week.